progeny is going to be called as the hybrid varieties. These progenies will not called as the transgenic animals, because the genes what you are introducing into this particular organisms are coming from the similar kind of species. For example, if this is a rabbit, this is also a rabbit, but this could be a rabbit with the some definite phenotype. So because of that, the progeny what you are going to get is actually going to have the properties from the male as well as the female, and that's how this progeny is going to be of improved varieties or improved characteristics. But these progeny will not going to carry any gene from the trans species. It is going to contain all the gene from the cis species, which means it is going to contain the DNA from the same species, and that's why these kind of hybrids are not called as the transgenic animals. Compared to that, you can be able to generate a transgenic animal with the help of the genetic engineering. If you recall, while we were discussing about the biotechnology principle and processes, we have discussed about how you can be able to generate the chimeric DNA. So what you have to do is you have to first isolate a genome, and then from the genome you have to isolate the gene of your interest, and then the gene of interest has to be digested with the restriction enzymes. Similarly, you have to process the vectors, and then you are going to put them together for the ligation reaction, and that's how you are actually going to get the chimeric DNA. This chimeric DNA then has to be introduced into the organism of your choice, and now at this stage this particular DNA either can remain within the cytosol. For example, in this case, when we are introducing this into the bacteria, it is actually going to give you the transformed bacteria. But there are other cases. So this particular type of bacteria will not be called, so it's going to have carry the gene from the foreign organisms, but it is not going to be a permanent species, because after the few cycles this plasmid is going to be removed, for example. And the other case could be that you actually going to clone the DNA into a vector, then you introduce this vector DNA into the organisms. Once the DNA will enter into the nucleus, so at this stage, once you enter the DNA into the organism, it could have two choices. One, it is actually can go into the cytosol, like it is actually can go into the cytosol, or it can go into the nucleus. So if it is goes into the cytosol, it will remain there and it will express for some time, but afterwards it will not going to integrate into the genome, because and it will be going to be removed. So these kind of organism where you are going to introduce the DNA into the cytosol are called as the temporary transgenic animals, or these are actually going to be called as the ectopic expression models. And the places where you are actually going to introduce the DNA into the nucleus, the DNA will enter into the nucleus, and then there will be an integration of the foreign DNA into the genome, and because of that, the DNA, the organism is going to be now permanently modified. So organism genome is going to be permanently modified in this particular case, and that's how these particular type of genome can be, or these kind of organisms are called as the transgenic animals. Let's see how you can be able to develop a transgenic animals. So if you want to develop a transgenic animals, you have to perform the 4 steps. In the step 1, you have to construct a transgene, which means the gene of foreign gene which you are going to interested into to introduce into the animals. So this transgene has to be cloned into a typical type of vectors, so that it should be able to integrate into the genome of the animal. Then in the step 2, you are going to introduce the transgene into the animal. So that is also has the multiple options which you can explore depending on the type of animal cell, and as well as the type of the vector, as well as the transgene what you are using. Then the third is, once you have done that, then you have to do the screening of transgenic animals, so that you can be able to screen the animals which have received, or which have received the gene and which have received the DNA into their nucleus. So at this stage, you can actually screen the organism for whether the gene is in the nucleus or whether the gene is in the cytosol. Because the organism, if the organism is going to have the gene into the cytosol, it is not going to be converted into the transgenic animals for the future biotechnology applications. So in the step 1, you are going to do a construction of a transgene. So the transgene is going to have the multiple components. Like the first component is that you have to isolate the gene of your interest. So gene of, as I foreign gene which you are going to express. So in that case, you have to first isolate the genomic DNA, and then from with the help of the PCR, you can be able to express or you can be able to isolate the gene of your interest from the isolated genomic DNA, which means first you have to isolate the genomic DNA, and then with the help of the PCR, you can be able to do the PCR of that particular gene, and you can isolate that gene in the large quantity. And then you require the vector DNA. So in this case, you don't need the cloning vector, you require an expression vector, so that it should have also the cassettes which actually can allow the expression of this particular gene of your interest. And then you require the promoter as well as the termination sequence. So this is a typical gene cassette which you can use for construction of a transgene, where this is the region which you are actually going, where you are going to put your transgene, the foreign gene. Then it is going to be flanked on the promoter side. So on the 5' side, it is going to have the promoters. This promoter is actually going to be corresponding to that particular animal. For example, if I am using a mammalian system, then this promoter should be going to be recognized by the mammalian RNA polymerase and mammalian system. And then following that, it is going to have the vector DNA, and on the 3' side, it is going to have the termination sequence, so that whatever the protein is going to be produced, it is actually going to be terminated. And following these promoter as well as the termination, you are going to have the vector DNA. And the scheme through which you can be able to do the construction of transgene is very simple. First, you have to take the genome of that particular foreign DNA. For example, if you are interested the cloning of the transgene of a particular gene from the human, then first you have to do is you have to isolate the human genome. Then with the help of the PCR as well as the primers and all that, that is what we have discussed when we were discussing about the biotechnology. You can be able to do the PCR of this particular gene. Then you do the stationarization and generate the cohesive ends. Similarly, you can actually be able to modify the vectors with the help of stationarization. You can generate the cohesive ends, and then with the help of the ligation, you can be able to generate the chimeric DNA. Now, this chimeric DNA is ready to be inserted into the particular animals, and that is the step 2, where you are actually going to introduce the transgene into the animal of your choice. So now in the step 2, you are going to introduce the transgene into the animal. So there are 3 methods through which you can be able to introduce the transgene into an animal. One is the retroviral vector based, which means you are actually going to clone the transgene into a retroviral vector. So retroviral means the viral based vectors. And then what you have to do is you have to take the animal and give the infection with this particular type of viral particles. And once there will be a viral infection into the animal, the virus is a natural, under the natural process, is actually going to insert the DNA into the cell, and then this DNA will go to the nucleus of the particular cell, and then it is actually going to integrate into the genome of that particular organisms. The second is that you can use the DNA microinjection method. So in a DNA microinjection method, what you are doing is you're taking a cell, and then with the help of the microinjections, which microinjections are like a syringe, micro syringes, where you can actually take this particular type of DNA, and then directly you can inject that DNA into the nucleus of this particular cell, and that's how you can actually be able to deliver the DNA into the cell. And once it reaches to into the nucleus of that particular cell, then it will integrate along with the genome, and ultimately it is going to proliferate along with these cells. The third is that you can actually use the embryonic stem cell mediated transfer. So in that case, what you can do is you can just take the embryonic stem cells and then you introduce the DNA of your choice, and that's how the embryonic stem cell can be differentiated into the different types of cells, and then you can implant those cells into the animal, and that's how you can be able to develop the transgenic animals. Now, once you have introduced the gene, you are actually going to have the two different species. One, the where you have introduced the DNA, but the DNA is remain outside. It does not go into the genome, or the inside the nucleus. The other species could be that where you have the nucleus and the DNA is within the genome. Now, this is the false positive, whereas this is the thing. These are the organism what you are looking for. So the next step is that you have to do the screening of the transgene into the animal. Now, if you want to understand the process through which the DNA will integrate into the genome, you can imagine that a cell has the this is typical animal cell. So what you have is you have the plasma membrane, you have all the membrane bound organelles, and then within that you also have the nucleus. So once it enters into the nucleus, it is going to encounter the genome. Now within the genome, it is not going to randomly being put into any place. For example, with outside the vector DNA, you can also have the integration sites. So you can also put some of the integration sites. So these integration sites are nothing but they are actually going to have the similarity to the sequence what is present onto this particular genome. So you can imagine that I am putting integration sequences of this flanking sequence of this particular genome. So this is a complete genome, and I have taken few sequence of this particular area and the few sequence from this area. So you can imagine that this is a particular type of gene which is flanked by the promoter as well as the terminator, and then I have just taken this particular flanking sequences. So what happen is that when this foreign DNA will enter, so you can imagine that this is my transgene. So once the transgene will enter, it is going to attach to this particular DNA with the help of this particular type of homologous sequences. So homologous sequences are the integration sites which are present on the both sides, and once they will attach to this, they are actually going to be replaced this particular stage of DNA with this particular type of DNA, and that's how this DNA is going to proliferate into the subsequent cycle. So in the first cycle, this will go and fit, and then the subsequent cycle, when there will be a replication, this portion is going to be removed and this portion is going to be replicated, and that's how in the beginning the two DNA will stick to this particular portion, and then later on during the cell cycle as well as the replications, this whole DNA is going to be replaced by this particular type of cassette, and that's how this portion of the genome is now being modified and where your transgene is expressing. So this transgene is now going to express and it is going to replicate, it is going to proliferate along with these cells as long as these cells. So if you do these kind of modification into the embryo or the zygote or all type of genetic cells, then this will continue even for several generations, because then whatever the organism you are going to generate, it is actually going to have the modified genome rather than this original genome. So that is the way the foreign DNA get integrated into the nucleus, and it get proliferated along with the several generations. Now, once you have introduced the DNA into the organisms, then you have to choose whether the DNA is being integrated into the right place, because it could be possible that it may find some similarity at this region and it could integrate into this region, but this region is not good enough because this region is not probably going to give you the expression. This region could be lethal for the organism, because sometimes if it can integrate it into a wrong place, then it might have, because once it integrated into this region, this particular type of DNA is going to be removed. And that's how this DNA could be essential for this particular cell, and that's how it may actually doing the malfunctioning. So that's why you have to do the screening of the introduced DNA, the organism where you have introduced the DNA, for two purposes. One, whether the introduction of DNA would be at the right place, number one. Number two, whether the DNA is being introduced and it is actually expressing and giving you the desirable traits or not. So that you are actually going to do by the simple PCR method. So you are going to do the screening of transgenic animals. So transgenic progenies are screened by the PCR to examine the site of incorporation of the gene. So you are actually going to go for this particular type of cassettes. In this particular PCR, you have the choices. Either you use the integration sites, or you can use some of the marker enzymes which you have put into this particular type of cassettes, or sometimes the gene of your place where you have actually introduced the DNA can be used. So there are three options. One, you can actually use this particular gene, and you can say whether this gene is whether you are getting a PCR of this gene or not, because this gene is not going to be present in this particular animal. So if you use the primers and you overexpress the primers, then if you are getting the PCR product, this means that the gene is being integrated into the genome. So that is the first question. It will say, okay, the gene is being integrated into the genome. Number two, you can use the marker enzymes. So you can, if you use the marker enzyme like thymidylate or some other kind of marker enzymes, that will actually say whether the integration is occurring at the right place or not. Or sometimes the third is that you can actually use the place where you have introduced the gene, and you can take the flanking sequences and do the PCR, and that will say whether the integration occurs at the right place or not. Because if it happens somewhere else, then that may interfere into the normal physiology of that particular animal as well. Now in the step 4, the animal breeding is to be done to obtain the maximal expressions. Now at this stage, you have the two choices depending on the type of the foreign gene you are expressing. Because if your gene is problematic, for example, if your gene is the lethal, or suppose the removal of this particular gene from the both chromosome is problematic, then in those cases what people do is they are actually generating the heterozygous offspring, which means only the one chromosome is having the mutations or having the introduction of the gene, whereas the paired chromosome does not have the gene. So those kind of organisms or those kind of animals are called as the heterozygous offsprings. Now once you want the homozygous strain, so what you have to do is, for example, the plus minus, this is called as the heterozygous. So what I'll do is I'll take the 2 plus minus and I'm going to crossbreed, for example. So if I do the crossbreed, what I'll do, I'll get the plus plus and I'll get the minus minus, right? I'll get the plus minus also, right? So in this case, this is the pure strain where the gene of your interest is going to be present in both the chromosome. And this you can just generate instantly, because I said, gene is problematic to the organism. So you just instantly generate, do the experiments, and then you finish up. But you always keep the organisms in a heterozygous condition so that the organism is going to be proliferate. But if you want to have the organisms where you want to do the homozygous, then you can actually be able to just simply do the mating for one generation. It is going to give you the single generation of homozygous strains, and that's how you can use this homozygous strain for whatever the exclusive purpose for which you are generating these transgenic animals. Now, transgenic animals are being developed for a specific as well as the exclusive purposes, and that's why it is important to understand what is the purpose of these transgenic animals, and that you will understand if you go through with some of the classical examples of the transgenic animals. So one of the classical example is the superfish. So there are multiple fish type transgenic animals are being developed, and they are being done simply by different purposes. So for example, when the people have developed the superfish, so superfish is being developed because it is actually being going to give you the bigger fish. So you see, this is the superfish and this is the normal fish. So this is the transgenic fish and this is the normal fish. Then you can see the comparison. This fish is thin, this is small, whereas this fish is very large and very big and very fleshy. So why it is so? Because you are actually doing all this into edible fishes, because that will actually going to increase the growth as well as the size of these fishes. So if you increase the size and the growth of that particular fish, it is actually going to give you the more weight of these fishes, and that's why it is actually going to increase the product